So my paper is called AAV Express ECD4IG provides durable protection for multiple SHIV challenges, or in simpler terms, a novel HIV therapy that can potentially prevent HIV from binding to and infecting human cells. So before I get into the nitty-gritty, before I get into the data, I just want to talk a little bit about why this paper matters. So in 2013, around 35 million people were living with HIV/AIDS, and around 1.5 million people died from HIV/AIDS. So that's a lot of people, and the current treatments for HIV are not exactly the best. The current treatments are antiretrovirals, and the main problems with antiretrovirals are, first of all, HIV can develop resistance to them. So because they work inside already infected human cells, antiretrovirals work inside already infected human cells. If HIV finds a way to mutate and kind of work around these antiretroviral pathways, then HIV can become resistant to the antiretrovirals. Second of all, they're extremely expensive. They cost around $10,000 to $15,000 a year, these antiretroviral treatments. And that's a big deal because I think it's 90 to 95% of people who are living with HIV/AIDS are in developing or third world countries. And so they can't really afford expensive treatments like that. And third of all, antiretrovirals have several unpleasant side effects like nausea, diarrhea, vomiting, things like that. Can anyone tell me the difference between HIV and AIDS? Does anyone know?
HIV is a virus that actually infects the human body and the human cells, and AIDS is the result of extended exposure to HIV.
Exactly.
One of these I'm trying to talk to you about.
Yeah, that's good. So, if you didn't hear him, he said that HIV is a virus that infects you, and AIDS is essentially a result. So, because HIV targets human immune cells, after all of your human immune cells are kind of gone, you're left with AIDS, and AIDS is acquired immunodeficiency syndrome, and that's basically where you're kind of immunocompromised and you can't fight off normal infection. In kind of current events, the Bill and Melinda Gates Foundation recently donated $6 million, around $6 million to the lab that conducted this research because it's such promising research, and you're going to see in a couple minutes the research. So thank you, Mark, for showing me that page. So now I'm going to go into the molecular pathway behind HIV infection. Basically, what you need to know for this paper is that HIV binds to human cells with these two receptors called CD4 and CCR5, and those are the two receptors that this paper works on. After infecting human cells, after gaining entrance to human cells by binding to these two receptors, HIV inserts genetic code into our genome, so that whenever we express those genes that code for HIV, we basically perpetuate the infection. We make more HIV, and that's a big problem because once it's part of our DNA, once this genetic code is part of our DNA, it doesn't matter whether or not this virion is dead. We're still going to be creating more HIV, so you're going to be infected. So that's why it's important that this paper works on binding to human cells. So if we can prevent that, we can prevent a lot of bad things from happening. Are there any questions?
You think you could talk about where most current HIV treatments?
Sure. Yeah. So antiretrovirals work inside already infected human cells. So after the genetic code is inserted into after the genetic code is basically injected into human cells, the antiretrovirals that I was talking about earlier, they work inside of the already infected cell. So, it's a little bit harder to prevent infection from that point than it is to just prevent it altogether before HIV binds to human cells.
I think they're anti-reverse transcription.
Yeah, that's one kind of antiretroviral. But I'm not really going to talk about that because that's a little bit more complicated. So now I'm going to talk about how it works. So this treatment is a protein. It's a protein complex that's over here. I'm going to go into that in a second. But how it works is basically there's something called an AAV, an adeno-associated virus, and it's similar. Actually, no, it's not similar to HIV, but it does insert genetic information into our genomes. So it's non-pathogenic and it's also non-immunogenic. So that means we won't have any there won't be any adverse effects if we take this this drug, this therapy. So what it does is this virus will insert the genetic code that codes for this protein complex into our genome so that we can express this protein without having to constantly take a drug. So we won't need to be constantly taking drugs like with antiretrovirals. It's kind of a one-stop deal. So once you get the treatment, you're good for life. You're ready. So now I'm going to talk about this complex. Basically, how it works is there's this top part, this blue, and there's this bottom part that's red. And I'll explain what the gray part is in the middle in a second. But basically, the top part, the blue part, is a CD4 mimic. So that mimics the human receptor on immune cells. And the red part is the CCR5 mimic, which mimics the CCR5 receptor in human cells. So what that means is that instead of HIV binding to human cells, it'll bind to this protein complex. Does that make sense? Okay. So once it binds to this protein complex, the protein complex will neutralize the virion because the virion can no longer bind to human cells. And this middle gray part over here is a type of antibody. And what it does is after the protein complex binds to the HIV virus, it will signal the immune system and tell it that there's something wrong, that there's a virus present. Any questions?
So Arjun, when the IgG1 binds to your gene therapy, then the immune system, the individual's immune system is going to attack the virus? Is that
Yes.
what you're saying?
Yeah. So, well, the CD4 part and the CCR5 bind to the virus, but the IgG1 is what signals the immune system. And I understand how that can be kind of confusing because HIV attacks human immune cells. I'm not really sure about that. They didn't really go into that. But because this, I'm gonna show you in a second how this therapy can be used as kind of a proactive, before you're infected with HIV, if you get this treatment, then you can be protected. So at that point, before you're infected, you will have immune cells. Does that make sense? So like, if you'll before you're, it's kind of like a vaccine. So before you're infected, you'll get this treatment, so that when you come into contact with the HIV, you'll still have immune cells that can neutralize the virus.
Now, HIV tends to mutate at a relatively quick clip.
Yeah.
So is this really a broad base?
Yeah. That's one of the other important things, that this treatment is the receptors that this treatment works on are conserved throughout all versions of HIV, because if it's HIV, it's going to be using this pathway to infect human cells.
Thank you.
Are these receptors involved in like other cell-related stuff?
Yeah, so they're immune cells. So normally CD4 and CCR5 are used. They're used to signal the immune system that there's an invader in the immune system. So that's a normal function. Does that make sense?
Yeah.
Are you asking what happens when you block them because then it shuts down that pathway?
Okay, so it doesn't block. Okay, sorry, I didn't explain that very well, but it doesn't block the human receptor site. It binds to the HIV receptor. So it mimics this bottom part of the human immune cell. So instead of binding to the human immune cell, the virus will think that this is the human immune cell and bind to this instead.
Thank you.
Okay. Any other questions?
Yeah.
So what about things that would otherwise normally bind to the cells receptor? Would they be attracted to the one? Yeah, right. Like non-HIV viruses.
Yeah. So that's they didn't go into that in the paper, but that might be something that they want to look into. I'm not exactly sure whether or not that's the case. Okay. So now I'm going to go into the data a little bit. The first test they did was in cells. So what they did was they inoculated the cells with this treatment with either CD4IG or ECD4IG. The difference is just this this little gray part. So CD4IG doesn't have it. But they did test. What they did was they inoculated the cells with either CD4G or ECD4G, and then they exposed them to HIV. And they did that for different concentrations of the two therapies, and they tested the percent infection. So how much of the HIV genes were expressed. And you can see that the important takeaway is that all of the blue lines are below all of the red lines, which means that ECD4Ig works better than CD4Ig, and that at a concentration of about 1 microgram per milliliter, all the infection is completely eradicated in cells that were given ECD4Ig. Questions?
Can you just point out which line that is on there?
Yeah, so just like if you look, the majority of the blue lines, it's not any one. So the ECD4Ig are all the blue lines, and the regular CD4Ig are all the red lines. So ECD4Ig is generally more effective than CD4Ig.
Are the different shapes just different strains of HIV? Or?
They are, I believe so, yes.
Then how are they specifically like measuring the infection? Just
So they kind of linked the genes that code for HIV and the genes that code for luciferase, which is a phosphorescent protein. So whenever the genes were expressed for HIV, the genes also were expressed for luciferase, so that they measure blood levels.
I think we have over 100% infections.
That's a good question. That might be either error or I don't know if someone else knows the answer to that question.
Yeah.
are these already infected cells? I thought you said that.
So they were already given the treatment before they were infected. Yeah, so they were given the treatment and then they were infected with it.
So if this treatment is around your cleanup, does that mean that if I get the treatment, will my kids also have immunity to HIV?
I'm not sure. I would, if it works the way I think it works, and I'm not exactly sure, but I would think yes. Okay, so I'm gonna have to move on. If you have any questions, you can come up to me after the presentation is over. So then they moved on to mice. So what they did was they again gave 4 mice ECD4Ig 1 week before they were infected with HIV, and they left the control mice uninoculated. So the HIV challenge is essentially just exposure to HIV. So the first challenge, all 4 mice that were given the inoculation were exposed to HIV, and 5 of the 6 control mice were exposed to HIV. And you can see that all of the inoculated mice survived, and all of the control mice died. And this line is basically just one mouse that they left for the second challenge. So all of the mice that were initially the control mice that were exposed to HIV died. And the second challenge, same thing happened. All of the inoculated mice survived, and all of the control mice died. Questions? Okay. Then they moved on to macaques. Rhesus macaques are a type of monkey. This first graph is an IC50 value. So what that means is how effective the drug is, and this is just comparing CD4Ig to ECD4Ig. And the takeaway from this graph is that the average concentration required to neutralize 50% of the virus is significantly lower for ECD4Ig than CD4Ig. And that just means it's more potent in a sense. This second graph measures viral RNA. So they inoculated 4 rhesus macaques with ECD4Ig, and they left 4 controls 10 weeks before the experiment started. And then 10 weeks later, they exposed them all to HIV. And they measured viral RNA. So viral RNA is a measure of how infected the macaques are. So if there's more viral RNA, there's more virus, and the organism is more infected. Does that make sense? So the takeaway from this graph is that for all of the inoculated macaques, the blue line is at 10 to the first viral RNA copies. That's very little viral RNA in the blood. And this is, you have to remember also, this is a logarithmic scale, exponential scale. So that the control macaques have significantly more viral RNA. All the red lines are significantly higher than all the blue lines. And these are very little viral RNA in inoculated macaques. Does that make sense? Any questions? Okay. So this graph is similar to the survival graph in mice, except they measured the percent of macaques that were uninfected, and they exposed them to actually 6 SHIV challenges. So the takeaway from this graph is, again, all the inoculated macaques, the blue line, survived throughout the experiment, and all the control mice died. Or all the control macaques became infected. Sorry. Questions? So SHIV is just simian human immunodeficiency virus. It's the rhesus macaque version of HIV. So, in conclusion, ECD4IG is an effective treatment for HIV in mice and for SIV in rhesus macaques. They can bind to many different forms of pathogenic HIV, actually all of them if they are technically defined as HIV. The ECD4IG can potentially be both a pre- and post-exposure treatment for HIV. Some further research that they might want to look into is larger sample sizes, human clinical trials, which they're actually starting to do. I found out about, I think it was last week. They were given money to start clinical trials, and they might want to test to see whether elevated CD4 or CCR5 levels interfere with normal immune function. I think you asked that question, right, Jane? And so, does anyone know how this new HIV treatment might upset the current system in place? So, like, normally, right now, antiretrovirals are drugs that you need to take regularly. So,
Well, you mentioned that they were the antiretrovirals were expensive, whereas this was kind of like you're a one and done basically. So it's like you're not going to be like money wise, not going to be funding farmers in the same way that you would
Right.
antiretrovirals.
exactly.
Also, because it's a one and done treatment, it's only one treatment instead of like refilling pills every couple months. So.
Do you know if the current retrovirals are generic medicine or are they patented medicine?
Are they what?
Generic or patented? Because if they're generic, the profit margin shouldn't be that high, so it should tend not to be.
I'm not sure about that. That's a really good question. I can get back to you on that.
I mean, the originals are certainly they're out of patent phase, but the newer ones I don't think are.
Right. Okay. Any other questions?
How can it be a post-exposure treatment?
Okay, so there were a couple graphs. I put that in, but there were a couple graphs I can actually send you if you want that showed that they were given the mice and the macaques were given the treatment after they were exposed to HIV. I'm sorry I didn't put the graphs in here, but.
So, like, going off of her question, does it matter, like, T cell count, like, if you're still eligible for the gene therapy?
Of course, it will matter in terms. So, in the protein complex has this antibody, and that's kind of this Torres's point that if you don't have T cells or if you don't have certain immune cells, that at this point this complex is going to be, this middle gray part is going to be kind of useless, but it is still an effective drug, as you can see by the CD4IG portion of the graphs. Any other questions?
I might have missed this, but is this something that you would have to take on somewhat of a regular basis?
No, so the whole point of this is that it's in the genes that code for this protein complex are in an AAV, and so they're inserted into your genome so that you can express them without having to constantly take the drug.
Can I read it? So there's actually two things that I think are really, really interesting about this paper. And one is popular opinion. So, this is a virus treatment, right? So you're going to tell somebody that just got infected with AIDS or HIV that you're going to give them another virus, and ideally that is going to cure them, right? So how willing do you think people would be to do that? Just a show of hands. Show of hands, I think people would be very willing, medium willing, not willing. So very willing. Medium willing, skeptical, not willing. So Konstantin Smirnov, a dude at Rockefeller, actually came up with a what was that big anthrax therapy like 15 years ago that utilized viruses to block any danger. And so this thing has been around for 15 years. It's been tested. It's been in the United States, and the Department of Defense has shut it down pretty consistently until just recently.
It's like the same thing as vaccines, right?
And yeah, just like another point off of that, this is a type of gene therapy, and gene therapy has been very controversial because it's kind of like you're modifying, not exactly modifying, but you're adding genetic code to your genome, so that's kind of a controversial subject. So finally, thank you to the Journal Club team. You guys helped me a lot. And of course, thank you for getting me this slot after I got stuck in traffic for weeks.